All right, these notes are going over the Chapter 4, Section 4, DNA connection. We did a guided reading last Friday, and that guided reading check video will be in today's folder as well. But these set of notes are going through protein synthesis again, kind of briefly talking about the different types of RNA, and then introducing mutations and how they are affected by protein synthesis as well. So please make sure you have your copy of the notes, and then we will begin. So the genetic code is something that we need to understand, and how genes play a role in it. So the main function of genes is to control the production of proteins in an organism's cell. So then proteins, they help determine the size, shape, color, and other traits of an organism. So these genes determine what proteins have to be made and what they'll be used for. A gene is a section of DNA molecule that contains the information to code for one specific protein. If you remember, a gene is on a chromosome, and that contains our DNA and that genetic information. So the bases of the gene then are arranged in specific orders. If you remember those bases of DNA, the A, T, G, and C, those bases are arranged certain ways to create certain genes. A single gene on a chromosome then can contain anywhere from several hundred to a million or more of those bases lined up in a specific pattern. So the order of the nitrogen bases then, so that A, T, C, and G, forms the specific genetic code that specifies what type of protein is going to be produced. And so in a genetic code, a group of 3 of those DNA bases codes for one specific amino acid, and those amino acids then build and connect together to create that protein. So they form in what's called a triplet. So like that A, C, and G would be 3 of those bases that would create a specific amino acid, and then T, A, C would be another amino acid. So for example, the base sequence CGT, which is cytosine, guanine, and thymine, is always going to code for the amino acid alanine. Sometimes the sequence of bases there'll be multiple sequences for the same amino acid. There's only 20 amino acids, so those combinations of bases can sometimes create the same type of amino acid, but it depends on how it's read. So then that order of the 3 base code determines the order in which the amino acids are put together to form that specific protein. So there's endless possibilities. Then the production of proteins and this whole process for how the code is read and what amino acid is used is called protein synthesis. So during protein synthesis, the cell uses information from a gene on the chromosome and it produces the specific protein. And then this happens in the ribosomes, which are in the cytoplasm of a cell, which if you remember, the ribosomes are one of the organelles of the cells when we compared animal cells and plant cells, and ribosomes were known as the protein factories. So the process of protein synthesis is kind of in like a 2 or 3 step process, but there are more details. So before this can take place, the messenger must first carry the genetic code from the DNA inside the nucleus into the cytoplasm. So that DNA is in the nucleus, that is the control center, and it now has to get that information into the cytoplasm. So the genetic messenger is called ribonucleic acid, or RNA. So the different types of RNA that we have each have different purposes. So messenger RNA copies the coded message from the DNA in the nucleus, and then it carries that message to the ribosome in the cytoplasm. Then another type of RNA called transfer RNA carries those amino acids to the ribosome and reads the code and adds them to the protein. This is kind of just a little diagram process. So the first step for DNA, or for this process, protein synthesis, is that the DNA has to unzip and separate its two strands into two open strands. So then the messenger RNA leaves the nucleus and it enters the cytoplasm. And then once it gets into the cytoplasm, it matches or it attaches to a ribosome. And then that ribosome begins the process of providing the code so it understands what protein is going to be made. Now there are free floating bases in the cell as well, so those get added and those kind of build upon it too. But the third step then is the transfer RNA will go through the process and attach to messenger RNA. And those bases then read the message in that 3-letter code, and they determine what amino acid has to be created and added. So then the fourth step is that protein chain is going to continue to grow until all of the amino acids that it needs are added, and then it'll receive a stop code, and the ribosome will release that protein strand. So with protein synthesis, we do have issues which are called mutations. So mutations can cause a cell to produce an incorrect protein during protein synthesis. So then as a result of that, the organism's trait or the phenotype, what it looks like or what that trait looks like, is going to be different than what it should have been. So in a body cell, the mutation will not be passed on to the organism's offspring. Your body cells are just kind of unique and individual to you. But those reproductive cells or those sex cells are where that information can be passed to an offspring. So if a parent has that mutation, it could transfer over into those reproductive cells, and the offspring could end up with that mutation as well. So some mutations are the result of small changes in an organism's hereditary material, while other mutations occur when the chromosomes don't separate correctly during meiosis. Then, there are some mutations that are harmful to an organism. There are a few mutations that are helpful, and then there are some mutations that are just neutral. They don't really help the organism, they don't really hurt it as well. So the mutation is considered harmful or helpful based on what it does to the organism's survival and reproduction. So if it reduces the organism's chance for survival and reproduction, then it becomes harmful because it's no longer helping that organism live. But if it does help the organism live, then it's helpful. Mutations also depend on the organism's environment. So depending on where the organism lives, a mutation in the desert for an organism might not be a bad thing, but if that organism would move to the Arctic or the jungle, it might become harmful. So helpful mutations then improve the organism's chance for survival, and an example of that is there's certain bacteria that have antibiotic resistance. So those bacteria, instead of getting killed off, they are able to withstand the antibiotic, and they can continue to grow and live. So this is the end of your notes. Please make sure you get everything filled out. If you need to go back, please do that. The PowerPoint slides are in here as well. You're good to go.